So it's so nice to be able to meet with you today. So excited to learn more about your lab. Um, and yeah, welcome to the show. So I was thinking first we could start with introductions maybe. So I'm Annika. I'm going to be a junior in the fall, and I live in Seattle, Washington, and I'm just really interested about biomaterials, and I hope to one day work in research and possibly even cancer research. I feel like that would be super fun and impactful. But yeah, I'm just super interested in biomaterials, and I can't wait to talk to you today about your lab.
Yeah, actually, the University of Washington has a really strong biomaterials background. So I don't know if you had a chance to uh, look into what's going on there and stuff, but there's definitely a lot of people at the university who are really doing amazing things in the
Mhm.
area of biomaterials. So
Yeah.
you're lucky to kind of be at one of the yeah, definitely among all the universities in the U.S. It's really has a lot of strong people and stuff. But yeah, it's great to hear that younger people are into biomaterials. I had no idea what they were when I was your age. You're definitely well ahead of where I was a long
Yeah.
time ago. So
I feel like
yeah,
they're also like it's a very like emerging field. Like maybe like just like 20 years ago, like it didn't even exist almost, and like it's only like upcoming now because like technology is like mixing with biology, which I think is super cool, you know?
yeah, it's definitely come a long way. Like the actual history of biomaterials are interesting. Some people will take it back to like prehistoric times where they would try to make really kind of basic things, but a lot of it started. There's kind of been different eras of biomaterials. So back in World War II, they noticed that something like plexiglass is called something called polymethyl methacrylate is plexiglass, and they noticed that like generally if you get like a splinter under your skin, it turns red, and so back in the day, most things you implanted into the body, your body just flat out rejected, which is a problem. And they
Hmm.
noticed that polymethyl methacrylate, when soldiers that they used it for like windshields and stuff, would it shatter, the body didn't really reject it. And so up until like the 70s, they were really just trying to find materials that wouldn't cause your body to reject it. And then in the 70s and 80s, it started the idea of can we actually use it to kind of direct things. And in the 80s and 90s, that really came along. And in the past 20 years, there's been a huge explosion just as we understand the body better and stuff. And so the things they do now, like with reprogramming and things like that, are really just things that would have been crazy to think about 20 years ago and stuff. And so I definitely feel like it's an exciting time and stuff. And it's interesting how things cycle back because for a long time, people were trying to basically stop your body from rejecting it. And so the immune system played a very large role. And then for a while, we felt like we had conquered the immune system, so we didn't have to worry about it. But now immune stuff is really huge again because the immune system, which is very complicated, unfortunately, plays a large role in how the body does stuff. Even the immune system plays a huge role in cancer as well. And
Hmm.
so it's definitely immunomodulatory materials are really my lab doesn't do that, but they're really kind of at the what a lot of people are trying to study right now.
Yeah, and like I feel like the more advancements that like happen within like immunomodulating materials, like I feel like the implications from biomaterials could like expand like exponentially because like once you learn how to control the immune system, you can really like do anything, you know?
Yeah. And then they use biomaterials to try to control the immune system, both for regenerative applications, but then also if you want to, for therapies that are immunotherapies where you're trying to use immune cells to fight cancer and stuff, can we use biomaterials? And at some point, the line between a material and a molecule gets kind of blurred. Like if you have a big piece of plastic that's a material, but then and a small molecule drug is basically generally considered a drug. But when you start building larger and larger nanostructures and stuff, sometimes it's not exactly clear whether you have a molecule or a material and stuff. But
Hmm.
yeah, there's definitely a lot of people doing really cool stuff in these areas.
Hmm. No, for sure. And next, I kind of wanted to talk a little bit more about like you as like a researcher. Like, how did you get into biomaterials? How did you become like a professor? Like, because I hope to do that one day. Like, that's super cool. So, like, how did you do it?
Sure. So the answer is it was kind of some people like are like you and they've known from a young age that they want to be a professor. I just kind of went to college and then I thought I wanted to be a chemical engineer at first, but I didn't really like chemistry, which is strange. My lab does a lot of chemistry now. And then I went to the University of Florida and they had a highly ranked material science and engineering program. And I had no idea what that was. And so they gave me a free water bottle and said, you should join. And I found that to be a convincing argument. And so I joined. And then this is so I was in undergrad from 2001 to 2004, just to put things in perspective. And so Washington itself, I think, has had a bioengineering program for a very long time, but a lot of universities have not. So, like Lehigh, where I'm at right now, didn't start its bioengineering program until like 2004 or 2005, and the department didn't start until 2017. And
Mm-hmm.
so when I was at the University of Florida, biomedical engineering was not a thing that I could have majored in at the time. And so I did not major in it because that was not available to me. So I did biomaterials with, so I did materials with an emphasis in polymers, just because a lot of polymers are used for biomedical applications.
Mm-hmm.
And then I did a PhD at Northwestern, also in material science. And it's just a PhD seemed kind of fun. I kind of liked research and stuff. I wasn't set on being a professor, but living kind of in the Chicago area and then working in a lab seemed kind of like it'd be a fun way to spend a few years. And I had a great time. And then I did a postdoc in London just because it seemed like it might be fun to live in London for a few years, which it was also very fun. And then I kind
Mm-hmm.
of realized that, like, different people like different things. Some people like seeing their ideas translated, in which case working in industry is probably a better idea because it's definitely a huge push on like getting things into the clinic and stuff. And a lot of really cool research is done in industry, but for some basic science stuff, it can be easier to do that in academia because the ideas aren't going to be profitable in the next 5 or 10 years or so. So, yeah.
Oh, that makes sense. No, yeah. And how did you make that switch from like material science to like incorporating biology into it?
So the when it comes to research, major means kind of less than you would think. And so if
No.
you go to college, you'll get a degree in something. And so if we have a degree in bioengineering here at Lehigh, we need to set course restrictions. We have people who need to teach classes and things like that. So your department matters a lot. But you can kind of do research in whatever you want to do research in. And so to some extent, the same person, people will frequently switch departments. Like it's not very common at a university, but like, I could easily, if I were to leave Lehigh, work in a material science department or a chemical engineering department, all of that would be painful for teaching. And so to some extent, like, you have your research platform and often that can fit into multiple departments.
Oh, okay. Well, that's interesting. And like, that actually kind of ties in what I wanted to talk about because I was looking at your lab's website and it said that like, you guys like use like a multidisciplinary approach to like approach these biomaterials. So what do you exactly mean by that? Like, how is that different from like what other people might do? And like, what do you exactly mean by multidisciplinary?
Yeah, so we take techniques. So we're interested in, one downside of being a material scientist not doing bio stuff is that I have no idea what people know. I guess you're still in high school, but like,
Mm,
I didn't take any biology in undergrad. And so sometimes
oh,
it's hard to know what people do know. Um,
yeah.
but Yeah, but so the idea is that cells secrete these molecules called enzymes, which are like catalysts that catalyze reactions.
Hmm.
And then a class of them are called proteases, which are proteins that cleave proteins. And so we like them because we can, peptides are short protein fragments. And so we can incorporate them into our biomaterials. And the idea is that cells themselves will modify the biomaterials in some way. And so one benefit, but a lot of the techniques we use were developed by biologists. And so we're kind of taking things that were developed by biologists and using them kind of more in a biomedical because, biologists want to understand how the body works. But as a biomedical engineer, we're kind of more interested in how can we change things. And so sometimes the biologists develop techniques to answer questions about how the body works, and then we take these techniques and we use them to try to modify things. And so the way we use them is slightly different, but we kind of use things from different areas and stuff. And so we also do some kind of basic material science research and things like that. So sometimes different projects can kind of be in different areas.
No, that's super interesting. And I feel that like also like since you're not maybe like a major biology person, you could have like a new perspective on these things that like people like are like set like standards in like biology, because you have
Yeah.
perspective.
And I
That's
know just in general with innovation, it really helps to have people who are from an adjacent field, but not the same field because they can understand things, but they'll have a different perspective on how to do things. And so
Mhm.
biologists really want to understand, why do things happen, what's happening inside the body, which is a very good thing. But then engineers are like, how can we change this in some way? And so the specific questions they ask, specific questions we ask, what we're looking for can be different. And
Mhm.
so sometimes it's helpful to take the tools they develop and then just use them slightly differently and stuff.
Yeah, that's super cool. And like kind of like my idea with this show was kind of similar in that like I want to get like other like my fellow like peers and my fellow students like interested in biomaterials because like like us as like young kids might have new fresh perspectives on these like on this research, which I think would be super cool.
Yeah.
Um, what other so another question I had was kind of like what is like the like kind of recent research that your lab has been doing? Like what are like the recent projects you've been working on and like what are you excited about?
Yeah, so I guess there's a few different areas of stuff that we do. So some of it is trying to make cell responsive biomaterials. So can we, all your tissues have multiple cell types working together. And so one idea would be, can we make a gel that will respond differently to a cell that forms a blood vessel than it might a stem cell or a bone cell or something like that? So are there ways that we can, make biomaterials that can better mimic tissues by kind of giving a different environment to different cell types that are present inside the tissue. So oftentimes, just from a synthetic standpoint, it's easy to make something which it's easy to make something which is just kind of the same throughout. So you put you have a hydrogel, and the hydrogel is all the same. But if you need different cell types require different things, and that can be difficult to kind of balance everything together. So one of the main goals of the lab is to see if we can make something that can make different types of cells happy within the single biomaterial.
Mm.
And then the other is to see if we can kind of make cells happy by better mimicking tissues. And so, uh, how stiff your different tissues are varies throughout the body. And so, your bone tissue or cartilage tissue might be stiffer than your brain tissue. And so we're trying to get a better understanding of how do cells are sensitive to this. And so if you put cells on a stiff environment versus a weak, not a very compliant environment, they behave differently. So we're trying to kind of understand what exactly is it about the physical properties of the environment, like how stiff it is, what happens if you apply, a force on it and things like that. And then how do cells sense this, and what are ways that we can design, gels and biomaterials that better mimic human tissues.
What could you like use these mimicking biomaterials for? Like, could they be used for like therapeutic purposes or like research, or what could they be used for?
So it's kind of both. And so therapeutics really nice. One challenge with therapeutics is just that getting it into the clinic can be a very long process. And so there's kind of two areas. The first is to use it therapeutically. And so if you can, inject materials that, if generally if your body heals well, we don't need to have therapies. And so, if you break a bone and you're very young, it often heals very well. And so we don't need biomaterial therapies for this. It's if you're older and you have a large bone defect, you might need one. And so, times that the body stops healing, can we make biomaterials that kind of promote healing in different ways? And then the second is, if we want to understand how the body works or come up with new therapies, it helps if we can mimic things better in the lab. Doing a human clinical trial is incredibly expensive and very challenging. But at the same time, most cell culture is done kind of on flat pieces of plastic. And so whenever you look at kind of the images of like cell culture, it's always these dishes and these trays that have some liquid on top of them. But your body is not a flat piece of plastic. And so
Yeah.
the better we can mimic it inside of a lab. If we start testing out drugs or trying to figure out therapies, the better we can mimic the body inside the lab, the more effective we'll be at developing therapies.
Hmm. So then, when you guys are like doing your modeling experiments in the lab on these like flat pieces of plastic, are you able to exactly model what the body's reaction would be, or maybe are you modeling more specifically the cell's reaction to it?
So generally, the first one is like a single cell type, and like a lot of it's called in vitro. And so in
Hmm.
fancy science terms, in vivo is something that happens inside the body, and then in vitro is something that happens inside of a dish.
Hmm.
And so what we're trying to do in the beginning is just usually most cell culture is just one cell type on a flat piece of plastic inside of an incubator inside of a lab. And so what we do is we make squishy gels, and so we put the cells inside of a more three-dimensional environment, and we try to see how close can we get these squishy gels to kind of mimic actual human tissues. And so human tissues, are very, very complex. And so an open question is like, how complex does it need to be to model it? And it's
Hmm.
also hard once again, because when you culture cells inside of a dish, let's say you're culturing bone cells, there is no immune system, there is no everything else. And so it's always a question of like, how complicated do you want to make something? Um, and how complicated can you make something to kind of mimic the different aspects that are important for whatever you're looking at.
Yeah, no, that's super cool. And like, I feel like, yeah, like I would love, that sounds so cool. I would love to do that. And then next I kind of wanted to also talk about, I was reading your most recent research article, which was on your lab website, which is about peptide derivatization and like how that can lead to peptide diversification. And I read it and I understood it to the best of my ability, but I had some questions that I like, to like learn more. So maybe first, do you think you could explain kind of just like in simple terms, like summarize what was the premise of that paper and like what was your goal with with it?
Sure, I'll just say in general. So peptides are short protein fragments, and
Mhm.
so one benefit of, and so there's a lot of things that work well with this, and so that the human body, like all your proteins can do a crazy number of things, and so things like the strength of your muscles, the ability of your eyes to see color, like just when you see a lot of stuff, just a lot of things in biology happens. And what's crazy is that when you see in the lab, we can make all sorts of crazy chemical designs, but amino acids that are found in proteins have relatively simple structures. It's not like they have super exotic with crazy elements. You don't see like metals you've never heard of in there and stuff. It's almost all carbon, nitrogen, and oxygen,
Mhm.
and hydrogen. And so they work really well. And one nice thing about peptides also is that they can be made relatively easily in the lab. And so proteins can also be made somewhat easily, but proteins, if you want to make them in the lab, you have to have bacteria make them or other things. Whereas, it's actually really simple. We've had high school students who can come into lab and start making peptides because it's relatively simple. One thing that you can try to do is to basically try to see are there ways that we can expand the chemical space for some of these. And so peptides, some of them are able to assemble. And so self-assembly is something where if you put a molecule in solution, molecules will aggregate and kind of form well-defined structures. And so generally, if you think of dissolving something in water, all you see is individual molecules floating around. But if you think of something like a cell membrane, what you have is basically the cell membrane is made up of phospholipids, and so you have something that has two different parts, and when you put it together, it forms a bilayer that wraps your cells. Peptides can do things like this as well. And so, to better understand how things assemble, sometimes you can kind of come up with relatively simple ways that we can derivatize these peptides in different ways after they've been synthesized in order to generate a large library to kind of understand how do things kind of basically assemble in order to see if we can make larger structures. And so this is nice. And it's one thing I kind of mentioned earlier about the line between a molecule and a material. With
Mhm.
self-assembling peptides, you make something that's very small, but it can assemble into larger structures. And so, it can be, depending on the state, you might say, oh, this is a molecule, or oh, this is a biomaterial, just depending on how it is, where small molecules kind of make up a material.
Mhm. So then when you're leading to the like deriving these peptides and like letting them self-assemble, is that mimicking the conditions inside the body in the cell that lead to these peptides self-assembling like in your body, or is it a different process which could create a new protein?
So this process is the way that it was used in the paper is slightly differently because it uses a chemistry that the body doesn't use for anything. But peptide assembly is actually very important because, and often for bad reasons. So there are cases where small peptides do assemble, but one case that's well known where it's not good, or cases are either some neurodegenerative diseases. So like Alzheimer's disease is due to peptide assembly into what are called beta amyloid plaques, which end up killing neurons and lead to the negative aspects of Alzheimer's disease. And so understanding peptide assemblies can be interesting, can be important for these because if you want to design drugs and things like that that slow down Alzheimer's, it'd be helpful if we had a better understanding of how things assemble. And the other one is what are called prion-based diseases. And so the most famous one is mad cow disease. And so these are a class of actually transmissible diseases, in which case one misfolded protein, which isn't exactly a peptide, but misfolded proteins can misfold other proteins and form structures that are kind of somewhat similar to the self-assembled peptide structures, where you have kind of these what are called beta amyloid regions that end up leading to assembly, but in these cases, the assembly is undesired.
Oh, okay. So then by achieving this molecular diversification, you're learning about all the different, like almost ways that a peptide can like misassemble, and so you can learn how to maybe prevent that. Or
Yeah. And then just in general, like peptide assembly can be useful. Just the lab went to grad school in Chicago was interested in making self-assembling peptides kind of for regenerative medicine. And so one nice thing about peptides is that if you put something inside the body, you don't want the body to react negatively to it. So if you do
Mhm.
have self-assembling peptides, one benefit is that you have peptides inside your body, and so your body knows how to break down peptides. And so if you take you can people come up with all sorts of great new molecules that the body's never seen before, but if your body hasn't seen it before, you don't really know how your body's going to react. Your body has just trillions of cells, and so you might test it out in culture and find that the cells in culture like it, but you put it inside the body and you find out that some population of neurons somewhere or some cell type in your body really doesn't like it. That ends up being a problem where you have negative side effects. And so one nice thing about peptides is that since your body knows how to handle them, peptides can definitely do bad things, but your body knows how to break them down and things like this. And so it can be easier. It can be somewhat less risky to use these within a biomedical application.
Yeah, and like the implications for like therapeutics also, because if you might not have a bad reaction from the body, then you could do whatever you want, really. Which
Yeah,
that's super cool. And then I was also, as I was reading your paper, I noticed that main method that you used for creating this peptide library is called Suzuki Miara coupling. I'm
yeah.
not exactly sure what that is. Do you think you could like expand on that and how it was important to your research?
Sure. So sometimes, it can be helpful if it can be helpful to have reactions that are very efficient. So just to be clear, it's a chemistry reaction, and so
Mm, yeah.
this is called a palladium catalyzed Suzuki Miara cross coupling reaction, and so the palladium catalyzed is helpful because it makes it work very well. And so if you have kind of a group called an aromatic group, it's a very selective reaction, and so um, just
Mhm.
in the field of chemistry, one challenging thing sometimes, and one thing that the body is really impressed with is if you do a reaction in the lab, you often will have, two reactants that come together and make a product. And so it needs to be very, very pure. And so the body is very different. Your body makes all sorts of proteins, your body makes DNA, your body makes all these things, but it's not like you have these compartments inside your body where you only have very pure things. The body is really, really good at doing chemistry in very impure environments. And so it's something that's actually very challenging for us to do as scientists trying to do things. And so one thing that's nice about reactions like the palladium catalyzed one is that there's not often a whole lot of side reactions. And so you can basically take a single depending on your chemical structure. But the reason that it was used for this is that if you have a specific moiety on there, so you have this aromatic group, and you can label this one, or you can react with a single position on there very specifically without having to worry about cross reactions across other things on the peptides. And so this is the palladium catalyzed one is not very friendly to biological things, and so we can do it in solution, but can't be done really in cells, or it's not optimized for what's called cells. But
Mhm.
recently there was something published. Recently the Nobel Prize was won for a class of reactions called click chemistries, which are relatively similar, where you can have very specific reactions between different functional groups. So you
Mhm.
can basically make a specific reaction happen, but even in the presence of cells. And so even if proteins have a bunch of different chemistries on them, nucleic acids have different chemistries, your lipids have different chemistries, but the click reactions, which were developed in part by Carolyn Bertozzi, who's on the West Coast down in California, won the Nobel Prize for making a suite of reactions that you can do kind of in the dirty presence of cells, where you have a whole bunch of different things going on. And so this isn't quite a click reaction the same way, but it's something that's used just so that, even if there's a lot of different chemical groups around, you can cause a specific reaction to happen.
That sounds so cool. Oh my gosh. Yeah, that sounds crazy. And then one last question I had about your paper was that in the conclusion, you kind of mentioned how this new chemical discovery tool can be used with machine learning and can possibly turn into like a computer-aided peptide design. Could you like expand more on this and kind of the implications of like how it can be used in conjunction with technology?
Sure. So like when we start doing when you add various when you have molecules in chemistry, they have they have different characteristics. And so, the reason oil and water don't mix is because oils have a lot of carbon, hydrogen, and carbon bonds, and these are non-polar bonds, and so oil is very non-polar, whereas water has oxygen, hydrogen bonds that are very polar. And so when we start functionalizing these peptides, we can say, we can classify these in different ways. We can say, how big is this chemical group? How polar is this functional group? We can find different ways to classify these groups, and then if we make a library of 40 different functional groups, we can look and see how do they assemble. And then the idea would be, can we take the different classifications, different ways to describe each of these, plug it into a machine learning algorithm, and then it can say, what, based on these different parameters, try to predict, okay, if we had something that looks slightly different, but was either bigger or more polar or stuff, we might be able to predict the way that things assemble. And so the idea is there could be an iterative process where you can easily functionalize peptides with a variety of different groups. We can then, basically use kind of machine learning algorithms to kind of analyze what is important about these groups that make these things assemble, and then feed that back in to say, okay, maybe if we try these 15 different functionalizations, they'll tell us something. So it's kind of an iterative process where we use kind of, we describe the different functional group or the different ways we modify it. We find different ways to characterize them and quantify them, feed it into a machine learning algorithm, and then see if we can then kind of use that as an iterative design process. And so there's a lot of different, and there's definitely, in the past 5 or 10 years, there's been, groups that have been at a variety of different universities who have taken really cool approaches to kind of, using automated synthesis techniques where robots actually make things, and then find ways to rapidly analyze stuff and see if we can, really automate a lot of the discovery process where we can, make it 1,000 different conditions, use machine learning to analyze what exactly is important, and then make 1,000 more conditions, use machine learning, and go through
Mhm.
millions of different permutations very quickly to identify kind of optimized structures.
No, yeah, that sounds, and like also like I'm actually working on writing and researching my first ever research paper, which is, and like
Oh,
I'm
nice.
using these like molecular docking softwares and like things like ChemDraw to like draw my own molecule and then see how it docks to the target protein. And I feel like that would be like a great like application for that. Um,
Yeah.
and then lastly, since I wanted to have a little bit of fun, this conversation has been a little bit like technical and serious. I had one last question for you, that I like to ask all my guests, which is if you could be a superhero and if you could have any superpowers in the whole world, what superpowers would you like to have to propel the biomaterial space forward? And like, what would you create with these superpowers?
Um, I'm trying to, there's a very specific, like, yeah, I guess it would be trying to think. It's an interesting question that I've never been asked before. So, yeah, like it'd obviously be nice if all experiments worked. One thing that is challenging when people first step in the lab is that, if you are in high school or college, if you study for exams, if you study for a lot for exams, obviously, like, I did bad on I got bad grades sometimes and things like that. But oftentimes there's a more linear line between working hard, and getting better grades, whereas in the lab, sometimes things just don't work and things like that. So it'd really be really nice if they could make all experiments work, just because it can be tough to work for a long time on something and find out at the end that for various reasons you didn't see before, things didn't work.
Hmm.
Yeah, if I could have the power of having my cells never get infected, that would also be really nice, just because it's tough for other reasons if you find out that there's bacteria growing inside your cells.
Hmm.
Um, yeah.
Yeah, no, you sound like a really cool superhero. Like
do
there would be
you have a
comic.
super do you
I,
have a superhero power for biomaterials?
I feel like one of my favorite superheroes is like Spider-Man. So maybe like I could like use his like web creating abilities to like create like some type of biosynthetic like hydrogel structure. That
Yeah.
would be that would be so cool. Um,
Yeah.
and then lastly, since I already kind of have you here and like you were so kind to like come on here and speak with me today and tell me about your research, I was wondering, do you have any like contacts or recommendations of who I could possibly talk to next, maybe at your own lab or your engineering department or just like anybody really? Because like I would love to keep learning about this type of research and this work.
Yeah, sure. I can. It might be easier to do that by email, so I'll reach out to some people quickly and see who's interested in stuff. But this is really great, and it's great to see people at a young age who are really interested in biomaterials and science and stuff. Because obviously, us in the field are very interested in this and think it's great. So it's really exciting to see that the next generation of people who'll be leading the field is kind of getting into it as well. So
Yeah, of course. No, that'd be so that I would, yeah, no, I want to be part of the field for sure. So yeah, that's super cool. And thank you so much for coming on here. This was super like insightful and I definitely learned a lot. I didn't know anything about peptide derivatization before, so I feel like this has been really nice to talk to you. And yeah, thank you so much.